So today, one of the biggest challenges in cancer is resistant to therapy. Tumor cells can escape the immune system thanks to an innate ability. They overexpress some of their receptors so that they can bind T cells and induce their exhaustion. But then what's T cell exhaustion? You can think about T cells as the killer sniper immune cells. That's indeed once the antigen, which is basically the enemy, is presented to them, they get activated, start proliferating, and kill their target. In an acute infection like influenza, for example, T cells effectively clear the enemy in around 7 days. And then once they've done their job, they just die, leaving only a pool of so-called memory cells, which are able to quickly mount an immune response if the antigen is presented again. But in chronic infections, something wrong happens. The virus persists, the T cells are constantly stimulated, and they go to a dysfunctional state called exhaustion. The idea of recent study is to characterize this hierarchical process with different stages, each with different functions. But still, there are a lot of open questions. What exactly triggers exhaustion, for example? Or what's the first symptom that we can observe in this process? Is this process reversible? And this latter question is particularly important in cancer immunotherapy and for pharmaceutical companies. Indeed, reversing this process and helping T cells in regaining their effector capacities can bring to very important outcomes for the patient. In my project, I would like to mathematically model this differentiation pathway toward exhaustion, as well as the heterogeneity in the process. Behind me, you can see a simple schema where represented in green the differentiation steps of a normal T cell differentiation pathway, whereas we have two different stages, red of the exhaustion, a terminal and a reversible one. Each state representing a subset of T cells is a variable and is described by an equation. So the interaction between the equation represents the interaction between the different subset of T cells. And altogether, my model can represent the dynamics of the whole process. For the future research, a collaboration between mathematicians and biology is extremely important. Indeed, as mathematicians, we can learn from the biology to build a realistic model, and then that can be then used to predict new results, test hypotheses, and carry on virtual experiments. Thank you.